It's my pleasure to introduce Professor Valérie Langlois. She's one of the world's leading experts in ecotoxicogenomics and endocrine disruption, which has been proven many times. And she has recently obtained a Tier 1 Canada Research Chair in 2024, has been appointed to the College of the Royal Society of Canada in 2023, and has received the Prix Relève Scientifique twice, namely by the Prix du Québec in 2020 and by the Université du Québec in 2019. So Professor Langlois is the chair of the scientific committee of the Fondation Evertea, we heard about earlier, which is located in France. She's the now outgoing president of the North American Society for Comparative Endocrinology and just hosted a very excellent meeting in Quebec City. She is the co-chair of Health Canada's Independent Scientific Advisory Committee on Pest Control Products, founded the Quebec Roundtable on Environmental DNA in 2025, and co-founded the International Intersectoral Center for the Analysis of Endocrine Disruptors in 2020. So she's been very busy recently. Professor Langlois also writes children's books to raise awareness about science and the environment among young audiences. And she does this, among other things, through her collection Professeur Planète et sa Brigade, which is published by Éditions Multimonde. In short, Professor Langlois has extensive expertise on the impact of organic contaminants on living organisms and ecosystems. And it's my great pleasure to introduce her. And Valérie will talk to us about a journey as a frog in an endocrine disrupting wetland.
Thank you so much, Jan. Oh, those introductions never make you comfortable. I need
I'll share the, sorry, give you access here.
screen.
Stop sharing. Okay, so you should be able to put up your presentation.
I share, and let's see if it works. Do you see the right screen?
Yeah, it looks great.
Okay. Well, thank you so much, Jan, for the introduction. Well, now that you said everything, I'm not gonna be I guess you won't be learning too much then. No, I'm just kidding. I guess the way that I'm allowed a bit more time than the student, I guess, because I'm older. So sorry, I cannot change the slide for some reason. Okay. Yes, okay. So because I'm a bit older, I get more time, but at the same time, I've been told that we are running out of time, so I'm gonna try to be quick. So I guess I split this talk in three. The first one, I guess, to show you my overall involvement in the EDC scientific community, sorry. Then more about the overview of the research program that we have in Quebec on EDC, and then like a specific example of like one project as an example. So let's get started with why, I guess, to try to prove to you that I'm worthy of this award. Hopefully, you're going to think that after the presentation. So as Jan just mentioned, I was the president of NASCI, which is the North American Society of Comparative Endocrinology in the last 2 years. And we just chair a very successful meeting here in Quebec City. So this group has been running for decades now, and I've been involved with many of my colleagues on participating in NASCI. And me and some of the colleagues here again, we are leading the EDC team. So we have to understand that for endocrine disruption, when we're talking about comparative endocrinology, it's all the other species but human. So we do have a lot of work to do, and it's very important for us to make sure that we keep do research on endocrine disruption at the non-human target. So that's what we are doing. As you know, obviously, I'm one of the co-founding director of ICEDA. So we launched this during the pandemics in 2020. So pandemics, I mean, everyone at home. So what could we do when we launch a network? Well, let's write like lit reviews. So many of you have participated in that. I think over 80 scientists across the world did participate in this collection of review papers that we push, I guess, as one of the initiative of ICEDA. So there were 14 scientific review paper on different topics that you can see here on the slide. I don't know if you see my little mouse here, but so everything's open access. Everything is available on the ICEDA website. So the only thing that is not open access is the editorial. For some reason, they don't put that open access, but it's kind of a nice one because it was really 25 years after the book was published, Our Stolen Future. I don't know if you read it, otherwise you should. I think it's where really the public got aware of the issue of EDCs, and I think that's very important. And I guess with this collection of review papers, we were able to kind of have an idea of like, how did we progress as an international society? So I encourage you to perhaps go and see those papers out. But here, what I like the most is like, it's been not even 3 years, and here the number of citation of the collection so far. So you can see that it was not just like a Canadian or Quebec, Canada, or like North American initiative, but really an initiative that is helping everyone. So this is a month old. So I'm sure now the data are updated and higher, but just to show to showcase the impact of the work we did. So this is what we can do when we are like 80, 100 people working all together on one team, which is the EDC. So I think this is a good example of the impact we have. So people that know me, I've been working on EDCs for quite some years now, and I've been we've been working a lot among ourselves, our researcher. We know it's important, but the public doesn't know. The regulators still don't believe it's really true. The industry is really good in putting this confusion in people's minds. So I think that was one of our mission, was really to basically take the conclusion of all those review papers and put them in 3 like kind of a public outreach piece that we put in the conversation. So I don't know if you know the conversation. I don't want to be spending too much time there, but it's really this journal online that all the scientists, master, PhD, postdoc, are actually the journalists of our own work. So it's kind of nice, and I encourage you to use it. So it can be in any language according if you have it in your country. So just so you know, these articles, those 3 articles have been read over 150,000 times, which is way better than any of my paper citation that I got in my career, right? So I think somewhere it's good to see the impact of this piece, and we don't know how far it can go. What I also realize, I do a lot of media work, I do a lot of public speaking, and what I realized was really that people that do listen to science or EDC or whatever, it's people that care already, people that know that is important, people that care about the environment, care about the impact of chemicals. But what about the other one, the other part of that population that is not interested? So I've been thinking a lot, and Isabelle and I wrote, like Jan said, the first 2 books on EDC, like the first one, because in my mind it was really important to try to go and get into the children's brain and put some seed on science, on environmental protection, to try to put those values from when they are younger, so hopefully it grows on them, because otherwise once you know, they reach the teen or adult age, it's perhaps not as it's more difficult to explain science, to give to talk about EDCs or any other environmental issues. And now, like Jan also mentioned, I started a collection with Multimonde, and it started on, it's only in French, so sorry for that, but hopefully it's keep, every year will be published one. And really the idea is to help the professors, the teachers and the students, the parents, you know, to talk about science. And so it goes, I guess, beyond EDCs, but it's really EDC that have started all this. So yes, we have to educate the public, but we also need to assist the government. So that's what I'm also doing. So this work was done with the government of Quebec, where, as you know, EDCs are coming from the wastewater, go to our lakes and rivers. We know that. What we are doing at the moment, we're doing analytical chemistry. We can see which are each of those chemicals. We know what's the limit of each of them, but you know, the truth for EDCs, as you know, it's the mixture. We saw that in the 2 other talks. We are exposed to a mixture of things. So here we're not in urine or in blood of human, but we are in the water that goes in the rivers, right? So fish, frogs, they're all like, you know, in that soup of contaminants. So working with the government, what we are doing is really creating those tests. So basically we're using the same assays that we seen just previously, androgen receptor transactivation assay, androgen receptor, we do anti or like pure, and then we look at is the mixture, right, regardless the components, is being an effect. If there is an effect, then we can go move into tier 2, try to understand why if needed. But really the idea is to find ways that could be a routine way of screening for EDC. So just so you know, we've been working with the government for the last 5 years, and now they have recruited my students, and now they are putting that in place. So I think slowly it's coming at you know, the regulation level. So that was my, I guess, more social part. So let's see the research program that I'm leading. I'm working, well, it's been a couple of years now that I have my lab, so I'm getting old. This is explaining all the white hairs. But so we've been working on many chemicals, many research questions, many things. But I pick one topic for you today. And the topic is the 5-alpha reductase. So many, when we started 15, 20 years ago, most of the EDC work was interested into the feminization, the increase in estradiol, effect of aromatase. You know, it was really, really female focused. So that's when, a long time ago, we started this to study the 5-alpha reductase, which is the enzyme. We go back to the enzyme that is actually converting testosterone to DHT, or 5-alpha dihydrotestosterone. So I know you know all of this, but you might not know that there's many types of the 5-alpha reductase, type 1, 2, and 3, and there's a beta form, and you know, it's a very complex thing. But the importance of those enzymes is very crucial in frogs. Its action, its activity will undergo like the reproductive activity of animals here, like we can see with this Brazilian frogs. Like I said, the 5-alpha reductase or the 5 reductase are very complicated. Here, just the androgen complex story, right? So we do have the testosterone and the DHT, but it's very, very complex. It gets degraded in many other metabolites. And even the DHT gets further metabolized into an androgen that's going to bind to the ER receptor. So, you know, it's pretty complex, all this. So it's very important to well understand this enzyme, to showcase that it's very important. We need to protect it. But what we discover throughout the years that it's actually one of the target of EDCs, which was not seen before the work we done. So I've been working with the frog Silurana xenopus tropicalis over the years. One of the reasons for that, it's you know, it's very easy to raise in the lab. Here you have our larvae and the frog, or the adult, sorry, stage, the genome sequence and the short generation times, and especially one of the main effect of the endocrine disruptors is actually the thyroid hormone disruption, which is a very leading the metamorphosis. So for us, we can see if there is like, you know, we can visually observe if there is an impact, okay? So here what I decided to do, just for the sake of this presentation, to just show you a bunch of EDCs that we've seen that it's actually targeting or affecting, I should say, 5-alpha reductase. So with some colleagues, always, and I thank all my students that are working on this. Obviously, I did nothing. I mean, they are the one running the show here. So here it's Laura that done this work. So really it was to understand the impact of arsenate. So arsenate, as you can imagine, it's a natural occurring environment. It's found both in developing countries. Here a map where you can see how it's contaminating drinking water across the world. So it's not, you know, you can see in the States, for example, there's many places where there's high contamination. One of the issue with high contamination of arsenic, it's been shown in human that has many effects. So effects on well, it's been linked to skin cancer, to lung cancer, to gallbladder cancer, to kidney cancer, many birth defects also, and several reproductive dysfunction that have been associated with exposure to high arsenate. So then we were like, okay, it's been affecting the reproduction. How come? So then what we decided is to expose the early embryo frogs to try to see, you know, what's the mechanism of action. So here, I'm not going to show everything we've done, but trust me, we've done a lot of genes related to steroid and thyroid hormone related genes that we analyze. But here, because we have the topic of 5-alpha reductase, we can see that we do see a decrease with concentration, and those are concentration that are actually found in those wells. So this is just to show that, okay, this enzyme or the expression of the enzyme has been one of the target. And that's kind of new because then you're like, okay, so not just the CYP19 is being affected, or so there's others. So it's very important. So that's a natural chemical, but what about the man-made one? Well, I don't see you right now, but I'm wondering who is wearing yellow at this point, because as you might know, there is some chemicals that are used obviously in the textile industry, and one of them is dispersed yellow 7. And what this chemical does, we don't know yet, but one of the project we were involved here, and it's Justine who led that project. But this is part of a bigger project with the Environment Canada, where we were looking at different outputs. But what was interesting, it's actually that so this chemical, you have to understand that it doesn't stay in the water column. It goes precipitate, goes in sediment. So we did expose embryos to sediment that were contaminated with the Y7, and we ran a microarray exposure. So remember, I'm old. Back in the days, we were still doing microarray exposure. Now it's more RNA-seq, but anyway, so you can see that it's really well split between the control and the Y7, and we look at many genes, etc. But really, I guess one of the key elements I want to show here, it's really, here we see an impact on the increase of the 5-alpha reductase type 2 again. So this is just to show that, again, we see effects on that one enzyme, and the expression of that one enzyme. So finasteride is another man-made chemical, but it's actually a drug, and it's a human steroid 5-alpha reductase type 2 inhibitor. So this is perfect for us to see what's happening in frogs, right? We use it for human against prostate cancer, because as you might know, when there is an overproduction of DHT, then you're more at risk to develop the prostate cancer. But if you block the enzyme or you reduce the activity, then you're going to decrease the DHT level. But then the problem of that is other people also, I guess, found that one of the reasons of the male pattern baldness, right? So the fact that people are, well, mostly men, are losing their hair is because they secrete, and it's the fault of the type 1 reductase that we have in the skin. that it's overproducing DHT. So basically, the men that are losing their hair is because they have too much androgen. They are too manly. So, but the problem that some of the, I guess, kids or more young men are doing now, they are taking pills of this drug so they don't lose their hair. So you can just imagine the impact that can have on their health. So anyway, I'm coming back to the main point here. So when we do expose our frogs to these drugs, here what we see a decrease of the same thing as we would have expected, right, in human and in frogs. But what's also interesting is we have the 5-beta 5-alpha reductase that, 5-beta reductase, sorry, that is also decreasing. So this is kind of new because none of the other studies back then had reported such an effect. Then when we do in situ IBE to the embryos, the frogs embryos, we see very similar things. So when we expose to finasteride, we can see that the control are, well expressed, but then the type 2, it's really almost no expression in the embryos, and very similar to the 5-beta. So there's still expression, but less. So anyway, this is just showing, I guess, over and over that yes, we see some chemicals can target this enzyme or the genes. So the plasticizer, it's another group of chemicals that you all know, and I'm not gonna be, I guess they need no introduction. And you know that most of them are either linked to the polymer or they are acting as a plasticizer. So it's more like oil if you want. So it's kind of a oil droplet between two polymer, and it's allowing the plastic to be soft and, to bind. So one of the problem is often when you have something new, it's like all oily and you're like, oh, this is a new car, it's so nice, but all this oil often is the plasticizer coming out. So there's many of them, as you know, and phthalates and bisphenol that we just saw earlier today are among the culprits or among them. So here it's a lit review that we published 10 years ago already. I'm getting old. And then what Justin did here is really to kind of collect all the mechanism of action that is out there for all animals of what those phthalates and bisphenols are doing into the cell. So we did a lot of work. Well, she did a lot of work and found many effects. But the one effect that obviously we are expert in the lab that was not yet done was to look at, what's happening on the androgen axes. But just so you know, because I think it's worth mentioning, but all the collection of these data show that these plasticizers do inhibit thyroid hormone. They do increase ROS, so reactive oxygen species. They inhibit cortisol level, estradiol, but again, like I said, nothing on androgens. So that's what we did here in frogs still in early embryo. This is Sonia who did this work. And then what we tested different phthalates in that case. Sorry, I don't think you see where when I'm moving my mouse on my other screen, I'm sorry. So we tested different phthalates. They are defined here, but the DEHP is one of the classic one known to actually being an EDC. But in our case, what we see an increase in all the reductases and some other effects in others. But again, just adding to the complexity of the, plastics and all the mechanistic that is surrounding. So then one question was like, okay, so all these work or all these effects, are they, directly related to, I don't know, androgen receptor and the androgens axis, or is it going through crosstalk? Because more and more we saw that when we had issues on the androgen side, we had also change in the thyroid word. So that's early work. Again, what we did here is to expose to T3, which is one of the thyroid hormone, and see if it would be changing the reductases level or the androgen receptor. And here what we saw, so really physiological concentration is actually increasing or affecting the androgen axis. So we know since years that there's a very classic crosstalk between the thyroid hormone and the estrogen, but less was known back in the days with the thyroid and the androgen. So more and more we were showing that actually there were like a crosstalk. So this is another paper. This is done by Diana, and then what we did, it's gonads that are directly exposed to T3, and what we see that in the testes there were no change, but when affected with T3 we see a decrease. So basically when there is T3, it's a negatively affecting the androgen. So then we were like, if we remove the T3, is it gonna lead to an increase? So that was one of the questions. So to test that, Diana, what she done was to actually use perchlorate. So perchlorate is one of the chemical that is used in bombs and all those explosive, but also in firework. And what we saw that with increased concentration of perchlorate, which is a known, sorry, I should have said it, it's a known thyroid hormone inhibitor, we saw an increase in the transcription. So there's really a mechanism of action that it's clear that's happening between the androgen and the thyroid. So this is just increasing what's difficult to understand when there's the impact of endocrine disruption. Is it, you know, directly to what we are looking at, or is it just like a consequence, you know, in a cascade of effects? So this is, I guess, one of the challenge of the field. So this is just to bring you back that when there is exposure to T3, we have a decrease in the reductase, and when there is an inhibition, we have an increase. So why do I care so much about all this? Because one of the consequences of affecting the 5-alpha reductase is totally screwing up the sex ratio. So when we do a chronic exposure to finasteride, to animals, then we have less males. So, and then we have these intersex that are coming. So intersex, it's really this mixture in the gonads or female and male cells. So I think this is and this phenotype also occur in humans, so you know, right? So it's important that we understand what is causing this and the mechanistic also behind. So it's one thing to find the endocrine disruptor, but it's really important to understand all the mechanistic behind this. So the road ahead, well, I guess what I've shown you for this part, the 5-alpha reductase, is that we've been using many chemicals, EDCs or inhibitors to try to understand, you know, the mechanistics. But I guess next, what's important to do now is to use knockouts to really remove those 5-alpha reductase to truly understand what's the impact, what's their role, and what's all the mechanistics that we were talking about. And stay tuned because Diana will be talking about it just after this. It's been years now that we are developing it. But trust me, it's a pretty long journey to work with knockouts, obviously. So I guess my last part of the talk will be also the role that have 5-alpha reductase in progesterone pathway. So I guess that's the, I will end with this study that we are running at the moment. It's almost done. We should be submitting it next week. So hopefully it's gonna, yeah, we'll see. Anyway, so the project I want to talk about, it's this idea that, you know, what's the role of progesterone in frogs and reproduction? Is there an impact of the reductases? You know, there are so many questions we need to address. So to work with that, we wanted to because just so I should say at the beginning, it's progesterone is known in mammalian species to be involved with pregnancy, right? But there's no pregnancy in frogs, but there's progesterone. So what's the role? So I guess it's really where we were trying to understand. So to study that, we went both ways with studying P4, so progesterone, but also using a progesterone or gestagen that is kind of a very, you can see the chemical structure. It's a mimic of progesterone. It's called melangestrol acetate, or MGA as short, and it's really used in the beef cattle industry, especially in the States, as one of the gestagen that they give as supplement. And they are using tons and, almost 300 tons of gestagen, sorry, that are just secreting every year, right? So there's a lot that are coming back to the environment. So the first, it's been years. I don't know if we see the year of those publications, 2020, 2021. So it's been many years ago that we did this work. And so we were interested in, what's the impact, what's the role and all that. So we did profile, we understood the effect. And that's the story I want to tell you today. Because, science is always this thing that you find things that you were not expecting. And I think this is a good story for that. So basically here the little, let me show you this. Here the little frogs. So what's happening, it's after the MGA exposure, there were no more legs or four limbs. So we were like, okay, wait a minute. That was not the reason why we, did all this work. We wanted to go mechanistically and crosstalk and all that. But then we also realized they were, missing legs. So we're like, okay, this is not normal. But the P4, the progesterone, exposure did nothing, but just the MGA. So we're like, okay, MGA is obviously doing something to those frogs. It's an endocrine disruption, something, but we don't know what it is. So this is where we decided to do another study to try to understand what's actually happening. And I should say that Paisley, who's the person that conducted the work, she just finished her PhD and moved into a postdoc position at NIH in the States. But Paisley, what she could see, it's like the arms of the frogs were there. They were just not coming out. So everything was very well, developed, but the skin, they could not go through the skin. So something was, not right, obviously. So here the research hypothesis that Paisley and Diana tested it in the lab in the last years. So what we know that in order for the arms to pop out, we need apoptosis, right? And this is controlled by thyroid hormone and glucocorticoid, right? But also, what we know is prolactin is actually inhibiting apoptosis. So we're like, okay, prolactin, how is that prolactin involved in frogs now? So now we're like trying also to understand the role of prolactin. And then what we also wanted to test is the fact, or we know, it's that prolactin and that glucocorticoid, what we know, it's they are working together in kind of skin remodeling. So to change the skin type between like the larvae to the adult, right? So those were the question we wanted to test. And I'm looking at the time, I still have 10 minutes. So I'm gonna try to go quick here. So here the experiment we did. So we redid the exposure, and we also got no arms coming out of the MGA treatment, which is good, to do a repeat experiment and get the same thing is always good. So we tested different treatment. So because to test our research question, we use MGA, and something called mifepristone or RU486. And really it's an anti-progesterone receptor, an anti-glucocortical receptor. We also tested MGA with metirapone. We always call it MTP. So, and that's an anti-cort, because we also thought that based on some literature that we read, that some studies have suggested that court could also sometimes create this phenotype. So we tested just court and an anti-court, and so we did all the treatment, but really we see nothing in the individual treatment here. So I'm going to focus today on the first 3 or 4 treatment with the control. So really what we see that's body mass. We see that, it's MGA is decreasing it. And then when we put it with the NT-PR and NT-GR, then it's kind of rescrewing it, right? It's significant. So it's rescrewing the phenotype. So that means we are in the right direction to find what's the mechanism of action. And we can see that when we get just the anti-court, that it's really not doing anything. So perhaps it's not really court related. Well, not as clearly anyway. Then we tested different other endpoints. So the developmental level, the length, the distance between the eyes. And then we always seen that MGA was decreasing it, and then a rescue, sorry, I should put my slide up, sorry, a rescue with RU486. And then this is kind of neat. That's Diana's work where she did histology on the actual legs. So I don't know if you can see the forearm here. This is the control. And what's important to see, it's the skin. So you can see it's very, very thin. And here we can see like a close-up. So it's really thin. And then this is MGA. Let me do this. MGA, which compared to it, it's really, it's thicker. You can see that the cells are different, and it's a very thicker membrane or skin type. Then when we put MTP with it, like we saw, it was no rescue. It's very similar in terms of skin. But then when we put the RU486, we can see that again the phenotype is rescued. So there's really something going on with those mechanism of action. So obviously we did conduct qPCR on several genes, and here's some of the results. So when we put MGA, we have increased, I don't see anything, I need my glasses. So when we put MGA, we can see an increase in prolactin. And for us, that was very curious or interesting. We're scientists. And also prolactin receptor was increased. And then we have VIP that decrease. We have CRH that decrease. And also thyroid-related genes decrease. So it makes it very complicated. So if we look at the CRH decrease, so that's in the brain. All those data are done in the brain of the frog. So what we decided is to use the tails to see what's the corticosterone, level. So we did that, and we saw like a very significant decrease when MGA is present. So we have kind of a rescue, but it's not statistic when there is RU486. But clearly we can see that at the hormonal level, that there's also a significant effect. When we look at the prolactin and also the thyroid hormone, then the fact that it's increasing the sensitivity of prolactin receptor, right, with the increase. So here, one of the hypotheses was that prolactin inhibit apoptosis. So it's possible that's what's happening in the MGA mechanism of action is that actually it's increasing prolactin, it's increasing its sensitivity. So basically it's inhibiting the apoptosis of those cells. But then at the same time, the very important balance between the thyroid hormone and the glucocorticoid is also affected. And then there's kind of a, let's say, the timing of the tissue for the cell proliferation for the adult, it's kind of starting at the same time, right? So it's really a complex crosstalk that it's happening. And really kind of the conclusion here, because of time, one thing is certain, it's MGA is affecting metamorphosis in frogs, because those little frogs, if they have no arms coming out, you know, there's no way they're gonna come out of the wetland. So they're like really, they're gonna die. But at the same time, as a scientist, it's always interesting for us to understand why. How is that possible? So really what we think is that MGA, by affecting this crosstalk, the very complex crosstalk between the thyroid hormone, the CORT, and the prolactin pathways, now that we are adding the three together, it's really affecting this skin remodeling to happen correctly. So, and I would like to end this by but adding support to two of my colleagues here, Laurent and Dan, that are suggesting years ago, that they have been suggesting that often we always think that thyroid hormones are the only one that are really involved in the metamorphosis of frogs. But I think this is once more showing that it's way more complex than just the thyroid hormone action that is actually driving the complete sequence of metamorphosis in frogs. So with that, I would like to thank you and also acknowledge all my students. As you can imagine, once you move out of as a student to a prof, you no longer are in the lab. So really, it's all the students' work that I've been showing to you today. And also it's because of them and the great team I had in the last years that that I've been able to do all this research. So thank you. Thanks to all of them. Thanks to ICEDA and all the funding agency. And with that, well, I think, yeah, I can take any question.
Thank you very much, Valérie, and congratulations again on the award. The floor is open for questions. If there are none right now, I'll start with one question for you, Valérie.
It's okay.
So yeah, I thought it was really interesting evidence you showed for the MGA and the mode of action and likely apoptotic sort of mechanisms. Are there any, did you think of doing any direct assays testing sort of apoptosis at specific stages or markers?
Yeah, well, you know, research is like this thing that you would never end. And that's something, you know, it's something Diana and I have often as a discussion is at one point we need to say, okay, this is it. We finish this and then we'll move forward. But yes, I think now that we just wrote the paper, and I like that I say I when it's Diana who did the work, but that we just finished the paper, there's so many questions and cool next step that we can do. I guess going into this apoptosis, it's not something that I guess we do think when we think of endocrine disruption. So I think it's opening another, like, you know, area of research that, yeah. But yeah, so to answer your question, we said that we'll finish other works before going back, but that's in our interest to go back to study more exactly the cells type and direct action.
Right, excellent, thank you. Vicky has a question as well.
Great talk, Valerie, congratulations. I was wondering a couple of things. The first was, you mentioned, you know, we often stumble upon these surprises and different modes of action and functions for different hormones. I'm just wondering how, you know, we're kind of pushing in vitro bioassays for monitoring for EDCs. So, you know, what are your recommendations in light of how complex and interactive the endocrine system is? How, you know, how do we sort of move forward with regulations and new testing regimes in light of that?
Yeah, well, it's a good question. It's a fair question. And I think I'm an advocate of doing both. I think there's place for both. I think when there's new chemicals or when it's for research, we have to use animal. We need to go to all system because it's so complex and we will miss so many things. This is perhaps one of the, you know, several examples we would have missed, right? But I think when we're dealing about regulation, because between the research and when we regulate, it's maybe 25, 30 years after, right? So I think we're pretty convinced that this chemical is doing something or this group of chemicals, then we can go in cell line you know, for routine assessment. For example, what I shown with wastewater treatment, if you want just to make sure that your wastewater treatment is effective, then you can just use cell line, because we're not there to, you know, learn new things. And well, if everything's dying, maybe we'll investigate, but you know what I mean? But when we're talking about new substances, you know what the government is doing, new substances or things that we know will have an impact, like pesticides, because they are meant to have an impact, we need to use the animal model. We need to do both, or like in silico and in, you know, all of them, but we cannot remove the in vivo model. And sometimes it's interesting to see why are we moving into the in vitro model. Who's pushing really so to remove the use of animal in research, right? It's interesting. You would be interested in the result. But anyway, my point is we should not stop using the animal model. I do agree we need to reduce. And when we are looking at the mechanism of action, like the other students are showing, they use cell line, they use this and that because they are interested in, you know, one tiny bit of the mechanism of action. But when we're talking about general impact, we need to use it. So that's what I will continue to say.
Great. I had one. Does anyone else have a question?
No, it does look like. Go ahead, Vicky.
She's frozen.
Oh,
she will be back.
yeah, we'll just wait to.
Hey, Juan. Juani is there. It's nice. It's like a family reunion.
I think Vicky's back.
Okay, Vicky's back.
Sorry, one quick question. Oh,
Okay.
go ahead.
No, go, Vicky. I think Juan, I just
Yeah, Vicky, go ahead, and then Juan.
Okay, so sorry, my computer froze there. I just wanted to address one other question, and that was most of your studies, or a lot of them, were done in Xenopus. And you were at Dr. Tyrone Hayes' talk at NASCI.
Yes.
The difference between lab-bred frog responses versus field-collected frog responses, very different. I just wonder if you think your Xenopus are reflective of other species. Do you plan to move into other species? Just your thoughts on that.
Yeah, it's a fair question. Yeah, I think what he shown, Tyrone, was that most of the lab line that we're buying are less responsive. So I think if we still see effects, I think we're fair, but I guess it's not a good answer from my behalf. We do work with also wild animal. I think what we've been doing in my lab and perhaps in your lab as well, it's the fact that we use multiple animals, different. We need to do because you know the variability between two different animals, vertebrate. If you see the same response, it's way more powerful than using another line of the same species. You know what I mean? So. And budget and this and that, and yes, if I was seeing nothing ever, I would change my frogs perhaps, but maybe you can argue maybe mine are too sensitive. I don't know. Yeah,
Thank you.
fair
Okay,
question.
thank you. Juan had a question as well, and then we'll move on to the final speaker.
Hi, everyone. Hi, Valerie. Very nice talk. Just a few short question. I'm surprised by it, but I know the impact for the when you read a text in the newspaper, that the impact in the general public is very, very high. Did you quantify what is the real impact, or how could be possible to quantify not only how many people read the article, if not the real impact? Because it's very nice. I amazing that I take your recommendation to make more write many news in the newspaper, because the impact in the general public is very, very nice.
Yeah, well, it's a fair question. It's difficult to answer that, and you know what? there's so many people. There's the thing is what I've been doing outreach for so many years, and I use different approach, like the media, the writing paper, the books, because it's never the same public. Some people will don't listen to the radio. Some people don't read, so I don't think you can just use one thing. So that's why I've been like, so for some of my colleagues, maybe I look like hyperactive because it seems that I'm doing so many things, but the truth is pretty much the same thing, just on another format, right? So I'm not too hyper. But to answer your question, your true question, that is, what's the impact? It's a tough one. We can see like what is the impact of on our work when it's been cited once in the paper. How can we answer that more than when there's 100 people who read your piece? Are people gonna change? I guess what we would need to do is to have a set public population. You put an ad, then you send a form to see to a survey to see if people learn, and I think it's social science kind of very nice project, and I'm pretty sure that some of our colleagues in the social world perhaps are doing that to try to see over their years are the overall environmental interest is growing. I would bet they do that, but exactly on one piece I write, I don't think it's it's that interesting or powerful when I write one thing. But I think it's a collection of effort. If I and all of you are talking about it, at one point, perhaps the population will be at least understanding the word disease or endocrinology, because most people don't even know what I mean. You know what I mean? So it's yeah, I think it's a lifelong project for me anyway. Thank you for the question.
Excellent. Thank you very much.